So let's study the structure of DNA. DNA is deoxyribonucleic acid. Now what is this deoxyribose? Deoxyribose is a sugar, pentose sugar. You have studied in your last class. Pentose, 5 carbon sugar, first carbon, second carbon, third carbon, and fourth and fifth carbon. Clear? These are, the fifth carbon has a phosphate group attached to it, and the first carbon has a base attached to it. Clear? Now why it is deoxyribose? Because here one oxygen is missing, hydrogen, polyhydroxy, these sugars are polyhydroxy, तो hydroxy में यहाँ O is missing. Clear? Now what are the bases? 4 bases: adenine, nitrogenous bases, they are adenine, thymine, cytosine, and guanine. Erwin Chargaff gave the rule that the ratio of purines and pyrimidines will always remain constant in DNA. The adenine and guanine are purines and thymine, cytosine are pyrimidines. Double cyclic or single cyclic structures की वजह से they are there. Adenine always pairs with thymine and cytosine always pair with guanine. Adenine और thymine में डबल होता है एंड इसमें ट्रिपल हाइड्रोजन बॉन्ड होता है. दे आर जॉइंड टुगेदर बाय हाइड्रोजन बॉन्ड. नाउ this whole is called a nucleotide. So what is a DNA? DNA is a polynucleotide chain. Polynucleotide means this kind of chemical structure is in the form of a chain. Say this is the chain. Okay, sugar, phosphate, sugar, phosphate, sugar, phosphate. Clear? Sugar, phosphate chain. There is one sugar, then one phosphate. These are attached with the help of bases. Adenine pairs with thymine, sugar is attached to base, thymine with adenine. Clear? Cytosine with guanine and guanine with cytosine. Koi doubt? So this looks like a ladder. Now this ladder is twisted. Adenine thymine में double bond. Twisted ladder like structure we will draw. See, you can draw with me so that you are able to draw it in your exams also, like a wave. Draw it, then parallel to this draw another line. This model of DNA was given by Watson and Crick model. This model is called the Watson and Crick model. These are the scientists who from X-ray diffraction studies analyze that this can only be the structure of DNA. It is in the shape of a ribbon, so draw a ribbon like structure here. Okay, now the other side also. In the exam, if you are asked to explain the structure of DNA, you have to draw this double helix. Imagine it in a 3D, means 3-dimensional structure it is. ये तो हम on a paper you can draw only 2D. So in 3D, these two helix, these two are ribbons are in the intertwined like a helix. This is double helical structure. Now in this, the 5 prime end of the sugar is this. In one chain, the 5 prime end of the sugar is on one side and the 3 prime end is on the other side. What is the meaning of this? This is the first carbon, first, second, third carbon. Third carbon hydroxyl is facing toward this side, and this is a fifth carbon जहाँ phosphate लगा हुआ है, that is the fifth carbon. So 1 2 3 4 5, this is the fifth carbon. Okay, so fifth carbon is facing towards one side and third carbon is facing on this, in this chain. Say this is the chain, say this is the chain. Whereas in this case, this chain will be inverted on the opposite side. So sugar will be inverted like this. Okay, so that the fifth carbon is on this side and third carbon is on this side. Okay, now the structure, coming back to the Watson and Crick model, this helical is twisted, and in these are the steps which are formed by sugar phosphate and these are formed by bases. Okay, so in this complete one turn, the distance is 3.4 nanometer, and between one base and the next base, in total in one turn there will be 10 base pairs. These are base pairs. So what will be the distance between one and the next? 0.34 nanometer. Clear? So, and the width is 2 nanometer. Width of the DNA is 2 nanometer. I hope you understood the model of DNA, Watson and Crick model of DNA. Clear? What does the Chargaff rule says? The purine and pyrimidine ratio will remain constant, means adenine will always pair with thymine and cytosine will always pair with guanine. Clear? So this is the structure of DNA. I hope you will be able to draw it. Thank you. In the next video we'll discuss the experiment to prove that DNA is the genetic material. Okay, thank you. So the whole summary of the discussion is here. DNA is a double helical structure given by Watson and Crick. Double helical structure में the backbone, it is like a twisted ladder. The steps of the ladder are bases and the sides are the sugar and phosphate. The two chains are anti-parallel, 5 3 end and 3 5 end. The two strands are joined by hydrogen bonds. Between the base pairs there is a hydrogen bond. Adenine pairs always pair with thymine. ATCG rule was given by Chargaff, and you can learn it by ATCG. You can call my professor used to say a tall charming girl. So you will always remember. The two strands are coiled in a right-handed helix, and the distance between 10 base pairs is 3.4 nanometer and one base pair distance is 0.3 nanometer. Thank you, and my next video will be on transforming experiment to prove that DNA is the genetic material. Thank you.